I think we can start. Hello, everyone. So, welcome to this STEM Quest session. Today, we will be talking about stem cells and how they can be very important for the future of regenerative medicine. And we will also look at some lobster simulation and how regenerating biology also works. So, we can start. And I have already been introduced a bit, but this is a bit about myself. So I'm a first year bachelor in biomedicine student at Karolinska Institutet, which is in Sweden. Um, I want to specialize in neuroscience later on after the third year, and I will conduct research on neurological disorders. In my free time, I'm a writer, I'm a violinist as well, and I like doing a lot of sports. So this is a bit about me. So yeah, let's jump right into it. So we will first be having an introduction on what stem cells are, what their properties are, and their potential. So that can then help us seeing how they can be applied into medicine and research specifically, because that's what we will be mainly talking about. And this will also help us answer the questions on the lobster. So then it gives a bit of an overview, and I hope I will cover everything extensively enough. But if there's any questions, I will. I hope I will be able to answer them. So, firstly, let's define what stem cells are. So, stem cells are unique cells which are capable of both self renewal and differentiation into various specialized cell types. They serve as a repair system to the body. So, they generate new cells for tissue maintenance and repair. I have added a small timeline which delves into what stem cells were important for in the last decades. And bear in mind, all of this research is pretty recent, so all of the ongoing research is still open and um, ongoing for the sake. So in 1981, the first embryonic stem cells were isolated. This was done by Martin Evans, which isolated stem cells from mice. And he established the foundational concept for pluripotent stem cells, which we'll be defining later. And he demonstrated that these cells could maintain an undifferentiated state in culture while retaining capacity to generate multiple lineages. So this was quite of a breakthrough in biology. In 2006, there was the discovery of induced pluripotent stem cells, which was done by Shinya Yamanaka, which reprogrammed adult fibroblasts into pluripotent stem cells using 4 transcription factors. He revolutionized the field by proving that cell identity is reversible and that patient specific pluripotent stem cells could be generated without ethical constraints. Why is this crucial? Because stem cells, which are pluripotent, can only be harvested from embryos, which is not only an ethical constraint, but also there are several limitations around research, whereas induced pluripotent stem cells can be generated, and then this results in an ethical approach to stem cells, which are pluripotent. And we will see what pluripotent means now. In 2000 to the present time, there have been a lot of clinical trials, and there has been this transition from the laboratory research to human trials. The first FDA approved induced pluripotent stem cell derived therapy for age related macular degeneration was just in 2023. So, from this, you can see how recent the research on induced pluripotent stem cells is, which is quite crucial because it means that everything is still open. So, first things first, when seeing at something in biology, you might have encountered that you always find properties. Why? Because properties allow us to understand why these things work, how these things work, and how they can be employed. So then one of the main properties of stem cells is indeed self-renewal. Self-renewal is the ability to divide and produce at least one daughter cell that retains stem cell identity over extended periods. So you may ask, then how are they different from any other type of cell? The key distinction from simple proliferation is that many cells can divide, but not all cells can maintain an undifferentiated state. So this is why stem cells are crucial, because they maintain their undifferentiated state as well. They don't only divide. So then the molecular basis of this is that they're maintained by transcription factor networks. and by signaling pathways such as the Notch pathway. The Notch signaling, just briefly, is a juxtacrine, so contact-dependent pathway in which a membrane-bound ligand on one cell binds the Notch receptor on a neighboring cell. So it's a specific type of signaling. So every type of stem cell communicates in different ways. And then, of course, the regulation of the cell cycle is important in this sense as well, which is regulated by the cycling CDK complexes, as you may already know. The second property of stem cells is potency. So the potency is the differentiation capacity of these stem cells. So we saw some terms related to this already, which is pluripotent. So the type of stem cells you can have differentiating are totipotent stem cells, which they can generate all cell types, including the extra embryonic tissues. And these are only the zygote and the early blastomeres. So these are totipotent stem cells. Pluripotent stem cells, which is what we already saw, are all the three germ layers. So the ectoderm, the mesoderm, and the endoderm. And these are embryonic stem cells, and now, with the current research, induced pluripotent stem cells. So you can see why pluripotent stem cells are the most important, because they give rise to the biggest amount of cell types. Then, after pluripotent, we have multipotent stem cells, which can give rise to multiple lineages within one tissue systems. So you can have blood cells, which are hematopoietic stem cells. Then you can have bone cartilage or fat. And then you have unipotent cells, which can just generate one cell type, and they retain some self-renewal capacity. So these can be, for example, skin progenitors or the hair follicle stem cells. Those just generate one type of cell. Then we have clonogenicity. Clonogenicity is a very important property because it's the capacity of a single stem cell to generate a clone of progeny capable of both self-renewal and differentiation. So in this case, we don't have regulation, we just have an assessment. So this is the main way to see if a cell is actually a stem cell. So how do you do this? You do this by colony forming unit assays. These are individually plated cells at low density, and what scientists do is that they analyze the resulting colonies. So the resulting colonies can be either mixed, so they can contain stem cells and undifferentiated cells, or they can be homogeneous and have only differentiated cells. Sorry. So which one is the actual stem cell culture? So the actual one is the mixed colony. So scientists, after they have done the assay, they look for the mixed colony. So those are the true stem cells, because it means that they not only differentiated, but they also maintained the original cells. So then progenitor cells only produced differentiated colonies, which will result in homogeneous assays. Then another important property is the quiescence of the cell and the activation. So this is basically means that the cells spend time in reversible cell cycle arrest. So they are not only active, and they can be active at different times in an organism's life. And this is maintained by the niche signals. What is a niche? A niche is a specialized microenvironment of supporting cells and soluble factors and mechanical cues, which is basically where the stem cells are lying. So then that's very important, because when a cell is taken out of the niche, when a stem cell is taken out of the niche, it's likely that it will differentiate. So it's very important that it's maintained in its environment. So the niche dependence is a crucial property of stem cells. And then we have epigenetic plasticity, which is not necessarily a property which is only of stem cells, but it's very important in every biological process. Why? Because this can regulate several processes. So it's the capacity to reversibly remodel chromatin and gene expression in response to differentiation cues, while retaining the ability to revert to the primitive state. So it's the ability of the cell to do different things, but also revert to the original state without having a permanent change. And this is done through pluripotency associated marks or differentiation associated marks. In the case of pluripotency, we have open chromatin at the pluripotency factors. So then it will increase the likelihood that that part of chromatin will be transcribed. And then we have differentiation associated changes, which means that this pluripotency genes will be silenced instead. So then it's an on and off switch mechanism in this case. So these are the main properties. So as we just saw, the stem cells can be broadly categorized based on their sources and differentiation potential, which makes them a crucial component in regenerative medicine and therapeutic applications. So what are the types of stem cells we can have? So these are a few of the types you can have, and they are the main ones used in research. So we already spoke a bit about the embryonic stem cells, which are the most researched and aimed because they are multipotent. So, pluripotent, and they are isolated from the inner cell mass of the blastocyst at 5-6 days post fertilization. So you can imagine that this is not the most ethical and non-invasive procedure. They are cultured on feeder layers or defined substrates, and their characteristics is mainly that they're pluripotent, so they can differentiate into all three germ layers, and they have a very high proliferative capacity. So then they can divide infinitely without senescence, without like having incurring into cell death. So they have the highest potency and they are best characterized. The only issue is that they are derived from human embryos. So there are moral objections in many countries, and several countries have regulations on them, so they cannot be accessed very easily. And they are highly restricted in many jurisdictions for the same reasoning. Then we have adult stem cells. Some of the cells I named under are part of adult stem cells. Adult stem cells are basically all of the somatic stem cells. So they are found in all adult tissues, which is bone marrow, adipose tissue, skin, muscle, intestinal epithelium, hair follicles, dental pulp, umbilical cord, blood. So it's all of the body tissues and where the stem cell niches are in these body tissues. And they are multipotent, so they are limited to specific lineages of tissues. Um, for example, the hematopoietic stem cells, they are used in blood and immune reconstruction and in leukemia treatment. So then they all have specific clinical applications according to the tissue they are found in. Which is very important. The only limitation is that they have a lower potency, of course, and they have a restricted potential compared to pluripotent stem cells. And they also have aging effects because they are taken from obviously somatic cells, which are in an adult organism. And this means that the proliferation and function can decline with age then. And you also have a factor of someone can or cannot be healthy, know it or not know it, so you never know what you encounter when taking stem cells because you have all of the environmental factors that affect someone's organism. Yes, then we have talked about the induced pluripotent stem cells, which, as we said, they are generated from somatic cells. And they are generated through forced expression of reprogramming factors, which are the original Yamanaka factors, which is the discover of the induced pluripotent stem cells. So then, in this case, they are equivalent to the embryonic stem cells in differentiation capacity, and they are also more ethical. They also have epigenetic memory, which is really interesting as they retain some memory of original cell type. This can be good, but it can also bias differentiation because then it can not be determined by chemical signals, but by this epigenetic memory that the stem cells have. So what are they used for? They are used for disease modeling. So they create induced pluripotent stem cells from patients with genetic diseases. For example, cystic fibrosis is a common one used. And then they also have unlimited supply, so they can generate as many cells as needed for the therapy. So you can imagine that they're really useful in regenerative medicine. So there have been some current clinical trials for induced pluripotent stem cells, and these have been done from Vertex Pharmaceuticals in the cardiac field. Then we have in Toronto and Mayo Clinic for the neurological stem cells, and for metabolic is still Vertex and JDRF. And for the retinal, this is Fate Therapeutics and Regeneron, just a few names. So this is what the ongoing research is about. Then we have the mesenchymal and stem stromal cells, which are found in the bone marrow, which is the classic source, but it's a bit of an invasive harvesting. They are also found in the adipose tissue, which is abundant and easier to obtain through liposuction or lipectomy, and in the umbilical cord blood, so the placenta and the dental pulp. And here they are taken in a less invasive procedure. They are highly accessible and they can be isolated and expanded relatively quickly. So they are still multipotent. So they differentiate into osteoblasts, which is the bone tissue, the chondrocytes, which is the cartilage, and the adipocytes, which is fat. Um, they have a very high plasticity, so they can undergo transdifferentiation, and we will see that later also in the lab simulation. Then we have a very important stem cell group, which is hematopoietic stem cells. Hematopoietic stem cells are basically all of the blood group cells, and they are really important because they are used for all of the main blood diseases. And those are really important because it's a large part of research. So they are taken from different areas. They can be taken from the bone marrow, which is the most classical surgical aspiration. Then you can have them from the peripheral blood, and in this case, they are mobilized from the umbilical cord blood, which is a non-invasive collection, but you get limited cell numbers, and from the fetal liver, which is a developmental source for hematopoietic stem cells. So the key characteristics is always that they're still multipotent, so they generate all blood and immune cell lineages, and they are maintained in the bone marrow niche by osteoblasts, endothelial cells, and stromal cells. So what are the clinical applications? The most established ones are on leukemia and lymphoma. Then there's also research on severe aplastic anemia, so with transplantation of hematopoietic stem cells, which can restore hematopoiesis. And then immunodeficiencies, which with gene therapy, which are approaches that are combined with transplantation of stem cells and gene therapy, and hereditary hemoglobinopathies. So even here you use gene editing and hematopoietic stem cells transplantation. So next we have the neural stem cells, which, as you can imagine, are in the neuroepithelium, neural tube, or developing brain ventricular zones. And in the adults, they're in the subventricular zone or lateral ventricles, and the hippocampal dentate gyrus, olfactory epithelium. So they're all the cells that give rise to the neural lineages. So they differentiate into all of the neurons and all the multiple subtypes, the astrocytes and the oligodendrocytes. So they're really crucial for spinal cord injury, Parkinson's disease, strokes, epilepsy, and traumatic brain injuries. So this is the main applications of the neural stem cells. Then we have the epithelial stem cells, which are found in the skin, the intestinal crypts, the hair follicles, the airway epithelium, and the corneal epithelium. They are multipotent to unipotent, so they have different ranges of potency. They can either differentiate in more types of cells or in only one, for example, the hair follicles. And they have asymmetric division as well. So the therapeutic applications are wound healing and burns, and chronic wounds and hair restoration, so and corneal disorders. Um, they are also used for intestinal regeneration, So this can improve healing after injuries or surgery through the application of these stem cells. Um, yes. So, as we saw, they play a critical role in regenerative medicine by facilitating the repair and regeneration of damaged tissues and organs. Their unique ability to differentiate into various cell types allows for targeted therapies, as we just saw, that can restore functionality to affected areas. So, this can revolutionize treatments in the future because they can first be um, tested in a lab environment. So we will delve a bit more on the applications of the different stem cells and for the specific ones. So I created a sort of recap. So as we saw, the treatment of blood disorders is one of the most important application. So leukemia and lymphoma, severe aplastic anemia. So the hematopoietic stem cell transplantation is what restores the immunodeficiencies with gene therapy and the hemoglobinopathies. So these are all the blood disorders, hereditary and non-hereditary. So these are really important because we have had many breakthroughs in gene therapy, but the combining gene therapy with the stem cell treatments can be really important because you can find a complete solution and not only a pattern into the genetic trees. Then we have the regeneration of heart tissue. So this is done through induced pluripotent stem cells. So cardiomyocytes are derived, and then mesenchymal stem cells, which we saw before, from the bone marrow and adipose tissue, and cardiac progenitor cells. So here it's a mix between harvested stem cells and induced pluripotent stem cells, which are engineered. Then we have the repairing of spinal cord injuries, and this is done through the neural stem progenitor cells, which differentiate into neurons, oligodendrocytes, and astrocytes to replace lost cells and remyelinate the axons. So this restores the injury. And then we have oligodendrocyte progenitor cells, which restore the myelin insulation on the demyelinated axons to improve the conduction and functional recovery. So this is really important. And the mesenchymal stem and stromal cells, which act via paracrine signaling to reduce the inflammation and modulate all of the glial scarring. So all of what concerns the neural stem cells in this case. Then we have the skin grafts. So the skin graft is when there is a burn victim and a different part of your body is taken, an epithelial layer, and it's placed on the injury you had. So where you have something that is like less useful for the body, you move it on the injury. And this can be done in three ways. So in the autologous split thickness skin grafts, you take the patient's own epidermis and part of the epidermis transplanted from a donor site to the burn wound for permanent coverage and good integration. Then for full thickness skin grafts, you take the entire epidermis and dermis moved from a donor site to areas needing more durable and less contractile coverage, for example, the face and the hands. And then for cultured epithelial autografts, you take keratinocytes, which are expanded in vitro in the lab, from a small biopsy to produce large sheets of epidermis for extensive burns with limited donor skin. So according to um, the intensity of the burn and the area that's needed to cover, then skin grafts are done accordingly. So this is more on the keratinocyte stem cells, melanocytes, or hair follicle progenitors side. So it's more of the epithelial stem cells. Okay, now we will move to the practical part of the session, which will include an overview on regeneration biology. So regeneration biology studies how organisms repair or replace damaged tissues and organs. And this ranges from a simple wound to complete limb or organ regrowth. It integrates developmental biology, stem cell biology, and tissue engineering to understand why some species regenerate extensively while others mostly scar. So, what is the difference in this case? We will try to understand what changes in different organisms. So, why is it that in some animals, for example, here we have the axolotl, but also in iguanas, if you cut one limb, it regenerates. But if we cut a finger, it's not that easy to regenerate, and often we have to resort to suturing. So, how would you define regeneration? Does anyone want to try to answer? So it's either scar formation, renewal of lost tissue, healing of wounds, and limb development. Does anyone want to try? Yes, you can also put the answers in the chat. Okay, yes, that is correct. It is renewal of lost tissue. So what we will try and understand is how the tissue is renewed and how it happens differently. So we will try generating some hypotheses. So for who doesn't know what Labster is, it's basically a virtual simulation in which you do all of the things that would happen in a lab. So I took some screenshots and I will take you through what it is. So we will try and design a hypothesis and then see what happens. So in this case, we will start with the axolots. Um, and in this case, we have an overview of the key procedure. And firstly, we have tissue regeneration. So what happens here? In this case, in the simulation, I took the animal and then I cut one of the limbs. And then he is first put into anesthesia. So the red bath is the anesthesia bath. He is put there and then it's speed up, and then you cut one of the limbs and you see what happens once the animal is awake. So it can take a few weeks. In this case, it was a sort of like 1 week and a half after to see what happens. So what happens, as you can see in the lower images, is that a sort of little mass of cells appears on the broken limb. And in this case, it's called a blastema. So we can view it on the microscope. And when the axolotl limb is amputated, what happens is that a blastema forms over the wound, and the cells inside then de-differentiate into multipotent stem cells that remain their positional information. So this is a light microscopy image of the blastema. The cross section was prepared 7 days after the limb amputation, and the blastema is coated by epidermis cells, which are stained in red, while the dotted blue line, the one on top, is the amputation plane. So it's where the cut occurred. And the area of unstructured tissues inside the blastema consists of de-differentiating cells. So then what happens is they try to make you hypothesize what would happen in humans. So I don't think anyone ever cuts their finger, but if your whole finger gets cut off, it won't regrow fully. And why is that? That is because regeneration of tissues occurs in different ways in different organisms. So why do you think? So on the upper side, we see an X-ray image of the finger. So in this case, the middle finger is the one that is most injured. And the hypothesis they're asking us to make is, why do you think the index finger was not sutured? So do you want to put the answer in the chat, or if someone wants to open the microphone and just take a guess between A, B, and C? So it's a hypothesis, so there isn't a right or wrong answer in this case. So it can be because suturing results in scars, because it prevents regeneration, because it results in enlarged extremities. So we have some B, some A, some B, mostly B. Okay, mostly B. So in this case, it's a correct hypothesis. It's a good hypothesis. There's nothing right or wrong. I would say that in the case of answer A, it does result in scars, but that may not be the problem, because the main issue when we are regenerating, it's not the scar, but the fact that the finger becomes functional again. So I would say B could be a good hypothesis, so that suturing prevents regeneration. So that is correct. And as regeneration does not work on large wounds, suturing is performed to prevent an ugly scarring. So then, if the finger was left to regenerate alone, it wouldn't regrow in a full functioning limb, let's say, as in the animal we used. But in case of the human, it would just form an ugly scar and it would be open and also open to infection. So then, suturing is performed in that sense because regeneration wouldn't occur fully and in the correct way. And we will see also why. So, yes. Now we will look at some microscopy. This is blastema cross section, and it's from different times. So the blue line, as we said, represents the amputation plane, and this is where the limb was originally cut. As you can see, the wound that was inflicted by the amputation is already covered by the blastema. So we have two blastemas, one on top and one at the bottom. Um, this is very important to see because you can see which one is the oldest. If you want to try sending in the chat which one you think is the oldest, if it's the one on top or the one at the bottom. And then we will see why it is. So one is like younger, it's taken at a earlier stage, and one is taken at a later stage in the images on the left. It's the same magnification, so it's seen under the microscope in the same exact way, and it's cut also in the same way, but one is younger and one is older. Okay, the bottom one, the one on top. Okay, so the older one, it's difficult to catch, but the older one is the one on top because the cells are rearranging already. So the enzymatic breakdown of the extracellular matrix results in a mix of different cells usually. And in the blastema at the top, this process is further advanced, and the cross section on the bottom was made days after amputation, whereas the one on top is a bit older. Um, sorry, a bit younger. Mm, yeah, so then you can see that the cells on top are on the bottom, sorry, are already regenerating. And this is the main difference between the two blastemas. So the cells inside the blastema, what happens when they are cut? They rearrange and de-differentiate. De-differentiation means that they change the course of the development and they give up their specialization. So then this is really important because they become specialized cell types and in normal development of stem cells. So when stem cells differentiate, they become specialized cell types, and here instead the reverse process is observed, which is really important. But then why does this reverse process not occur fully when we have, for example, a human finger? So we have a new hypothesis in this case, where the de-differentiated cells can only differentiate it into certain groups of tissues. So how was this tested with the obviously the animal organisms? So we can transplant tissue from the so firstly we genetically modify one of the organisms to express a green fluorescent protein, GFP, and then we can see them under UV light. So if you see in the bubble, there are two organisms. One is bright green, that is the green fluorescent protein organism. So it's just modified so that it expresses the protein. Then what we do is that we create a chimera. A chimera is not only a mythological animal, but also it's used in science to indicate an organism which is created by transplanting the tissue from the green fluorescent protein axolotl to the wild type axolotl. So what is done is that part of the limb, part of the skin is transferred to the other one so that you can see how the cells de-differentiate and with which intensity. So Before, we hypothesized that the de-differentiated cells could only differentiate into certain groups of tissues. So then, after a cross section of the limb was taken, um, what was seen is that the GFP can be found, so the green fluorescent protein can be found in skin cells as well as cartilage and nerves, but not in muscle cells. So then, the axolotl, which had the skin transplant, which was only on one part of the limb, he had a very light green expression of the protein, and it was mostly scattered. So it's not bright green as the first organism, which was genetically modified, but it was much lighter. So this means that not all groups of tissues were differentiated. So then it confirms the hypothesis we set. Um, yes. So what happened and why is that muscle tissue only develops from the mesoderm, not the ectoderm. So then the ectoderm is what forms the tissues as skin, nerves, and the epidermis. So then this is what was differentiated, but the muscle tissue cannot be differentiated. So then in the axolotl, when the limb is amputated in different areas, what is interesting is that the tissue regenerates in that case. However, we can conclude in this case that the cells maintain positional information while differentiating, which does not occur, for example, in the finger. And the differentiation is different and with different types of tissue. So, why is it important in the axolotl? It's important because when the limb is cut in different sections, so it can be cut firstly in an upper area and then in a lower area, the limb will be regenerated in the same way, which means that the cells maintain a sort of memory of where the positioning was, which is very interesting for future research. And it's a crucial point to take into consideration when you are transplanting stem cells between the different tissues and for future research. So that's where I sort of wanted to reach. That the use of stem cells is really important because it can show how the different applications can be made, but also how different discoveries are made because regeneration is different and the applications that stem cells have will be therefore different and across different organisms. So the way that axolotl regenerates their limbs is different than the way in which we regenerate our skin, which means that there will have to be differentiated stem cell treatments. So yeah, that's mostly it. And I also saw that the call is ending soon. So if you have any questions, I can answer. But otherwise, that was it from me. I will also put my email in the chat in case there's any additional question. Yes, and we can also share the presentation, and I have an additional one with more information, like a sort of a script with more information I can share in case someone wants to delve more on the theory part at the beginning. Thank you so much, Guy. What is the difference between labeled cells and stem cells? So labeled cells, it's mostly it's also based on how you can stain the cells. So when you want to label cells, you stain the cells basically. So it's all about what you are researching in that case. In all of biological processes, you do not always see the things that are occurring. So sometimes you recur to staining or like using antibodies to recognize cells. but mostly cells and stem cells, like the only difference between them is that the stem cells, as I said before, they have the ability to maintain their original state. So that's what is the main difference between them. But a stem and a stem cell and a cell are basically the same thing with that single difference. But that single difference is what makes the idea of regeneration possible. So I think that's like the main difference. I don't know if that answers the question. Thank you so much, guys.